Alright, so we're going to talk about the structure of DNA. Now, specifically, first we're going to talk about how DNA was discovered to be the source of genetic material. Alright, because that wasn't always the case, or at least what was thought to be the case. Alright, then we'll talk about how DNA is replicated, how we make copies of our chromosomes. Right, and we'll talk about some additional topics that tie into these processes. Alright, so first off, alright, so the early of 20th century, it's still kind of out there as to what was the source of genetic material. Is it DNA or is it protein? Alright, and most scientists at the time leaned more towards the idea that it was proteins. Alright, there are a few very innovative scientists who were rebels that challenged this notion. Alright, because if you think about it, logically, to them it made sense. Alright, because they knew that DNA was composed of nucleotides, and we know that DNA, there's 4 nucleotides, right? We have adenine, we have guanine, we have cytosine, and we have thymine. Alright, well, that's only 4 nucleotides. And at the time, they didn't know what DNA as a molecule looked like. Alright, they just know that DNA was made up of nucleotides. And they knew what nucleotides were composed of. Alright? And that proteins are made of these things called amino acids. And that we have 20 naturally occurring amino acids that occur. And so it made more sense to them that, hey, there's more different types of amino acids. So that leads to what they thought to be a higher degree of diversity. So that made proteins kind of the top tier as being the source of genetic material. At least originally, that was what was thought. And we all know that DNA is composed of, chromosomes are composed of DNA and proteins. Now, the proteins are histone proteins, alright? So the DNA molecule is spoiled around these histone proteins, alright? We've talked about chromosome structure previously, alright? So it's one of the two. So one of the first pioneers, all right, in the early 1900s was Frederick Griffith. All right, so he produced evidence that DNA was the source of genetic material. Now, even though he had this irrefutable evidence, all right, unsubstantiated evidence, the scientific community still said, not enough. We don't believe it. Alright, so, they kind of pushed his data aside and said, you're a rarity. Alright, so here, looking at his experiment, alright, it's brilliant. Alright, so you have 2 strains of Streptococcus pneumoniae. Alright, so this organism, this bacterium, causes pneumonia. Alright, so pneumococcus, that's another name for it, kind of the common name. And you have one strain that's virulent, meaning it's going to cause disease. It's pathogenic. And then you have another strain of the bacterium that's identical, except for the fact that it's avirulent. It's non-pathogenic. Alright, so he took this virulent strain and he exposed it to high heat and he killed it. And then he took the remnants of this heat-killed bacteria and mixed it with viable living bacteria of the avirulent type. And behold, he ended up with, at the end of the experiment, pathogenic strains of bacterial. Alright, it was magic. Alright, that here you take DNA and other components from this heat-killed bacterium and expose it to this non-pathogenic strain. And somehow this non-pathogenic strain is altered and becomes pathogenic. Alright, so he coined this process, transformation, because the non-pathogenic strain was transformed into a pathogenic strain. Alright, so let's look at the experiment in a little bit more detail. Alright, because he had some controls in place. Alright, so the hypothesis here is that the material in dead bacterial cells can genetically transform living bacterial cells. Now, what that source of material is, he's trying to figure that out. Alright, so here you have your solution of living Streptococcus pneumoniae that's virulent. Now, this living S strain, alright, S refers to smooth, alright, because it is encapsulated. Alright, it has a capsule, which is basically this extracellular polysaccharide protein gelatinaceous coating around the cell. Alright, and Streptococcus pneumoniae strains that are encapsulated are pathogenic. Alright, so this capsule is what we call a virulent factor. Alright, so he injects these bacteria into mice and they all die. Alright, so he found living strains of the bacteria in the heart of these mice. Alright, so here, looking at the living R strain. So this is the R strain is the non-virulent strain. It's the avirulent strain. R stands for rough because they are not encapsulated. Alright, so they lack a capsule. They lack this virulence factor. So injected into the mice, no bacteria are found in the heart. Alright, so the mice survive. Now, what happens when you take this pathogenic strain, this S strain, and you expose it to high heat? Well, that causes the bacteria to lyse. Alright? They die. Alright, so these guys here are dead. So you take the dead bacteria, inject it into the mice, and makes sense. The mice survive. Now, what happens if you take a heat-killed S strain, alright, and you toss in some living R strains, and then you inject it into the mice? Well, the mice died, and he found living S strain cells in the heart. So somehow your R strain was transformed into a S strain. Now, what caused that? All right, now, there's some transforming substance. So the question now becomes, is it DNA or is it protein? Or, here's another competing thought, is it RNA? Alright, so we got DNA, we got RNA, we got protein. Which is it? Well, here, we have our second experiment. Alright, so here, the chemical nature of the transforming substance from pneumococcus, alright, it's DNA. That's what Griffith is claiming, is that it's DNA. So, how do you go about proving this, or at least substantiating this claim? Alright, so here, you take your S strain, and you expose it to high heat. So then you have these bacteria that have lysed, and so then you homogenize them. So you put them in a blender, you make sure that your sample is completely homogeneous. You mix it up. And then you filter it. Alright, so all you're left with are the soluble components. Alright, so the actual cells themselves in the filter, they're too big to pass through the filter. So you're left with DNA, you're left with RNA, you're left with proteins. Okay. Now, not all proteins are going to pass through the filter. Okay, but you are going to have some short proteins that pass through. So then you split the sample into a couple of different test tubes. To one of the test tubes, you add an enzyme called RNase. Now, the RNase degrades RNA. Alright, so that means that all you're going to have in that sample is either protein or DNA. To another test tube, you add protease. Well, the protease destroys the protein, so you're left with either RNA or DNA. And then, in another test tube, you would add DNase, which destroys DNA. So you're left with RNA, and you're left with proteins. Alright, so then you transfer this solution to your rough strain bacteria. And you incubate the samples. And then you look for the presence of smooth strain cells, encapsulated cells. Alright, your transformed cells. Alright, that's what you're looking for here in your results. Alright, so the idea here is if the component that's responsible for serving as this transformation factor is destroyed, then your sample is only going to have rough strain bacteria, not smooth. Alright, so by treating your sample with RNase, you still have encapsulated bacteria. So RNA doesn't work. Alright, RNA is not the source of genetic material. Here, treating with a protease, alright, you end up with encapsulated cells. Well, that means that proteins are not the source of genetic material. Treating with a DNase, right? You have only rough strain bacteria, no smooth bacterial cells, no virulent cells. So that told Griffith that the source of genetic material is DNA. Alright, now, Frederick Griffith's experiment, or at least his results, were refuted. Alright, so here we have another experiment that was done that we actually give credence to as being the kind of big overall experiment that says, okay, DNA is a source of genetic information, and proteins and RNA aren't. Okay, so first we gotta talk about viruses, just a little bit. Alright, so here you're looking at a virus that's infecting a bacterial cell. Alright, so we call these phages or bacteriophages. Alright, so the virus. Alright, now viruses can have genetic material that's either DNA or RNA. And the virus itself, alright, what makes up the virus, the outer structure, is proteins. Alright, so your virus, your phage, has a little head. Okay. Alright, so your virus has a head. It's got a tail, and it's got little feet. It's got a little tail sheath, so it's got a neck, and it's got little feet that we call tail fibers. And basically, the head, the capsid, is where your genetic material is enclosed. It's inside this capsid, or the phage head. And the tail fibers actually attach to the surface receptors on bacterial cells. And then the phages will inject their genetic material into the bacterium. Alright, so this is different from most of your viruses that actually enter a host cell. Your phages don't when they infect a bacterial cell.
Bacteriophages, or phages, are viruses that infect bacteria. This is a T2 phage, which consists of DNA inside a protein coat. The reproductive cycle of the T2 phage begins when the tail fibers of the phage stick to the receptor sites on the surface of a host bacterium, such as E. coli. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The DNA of the host cell is destroyed, and host cell enzymes and nucleotides are commandeered to replicate the phage DNA, making more phage DNA. The host cell's enzymes and ribosomes are used to manufacture phage proteins. Phage parts accumulate and assemble to form phages. A phage enzyme digests the bacterial cell wall and the cell ruptures, or lyses. As many as 200 phages spill out. Each of them may go on to infect another cell. This diagram summarizes the reproductive cycle of bacteriophage T2.
Alright, so here you're looking at the life cycle of a lytic phage, like your T2 bacteriophage. Alright, so your phage attaches to the surface of a bacterial cell. It injects its genetic material into the host cell. This genetic material is then used by the bacterial cell, or the genetic material is going to be copied and be transcribed and translated into viral components, while host bacterial DNA is degraded. And then, lastly, the genetic material, the DNA, and all your little viral components, the tail fibers, the neck, and the head, the capsid, are all going to be assembled, and your little virions will eventually lyse and be freed from the bacterial cell and go on to infect other bacterial cells. Alright, so here we have our second experiment, 1950s. Alright, we have Hershey and Chase. Alright, so Hershey and Chase are the ones that are credited with the fact that DNA is labeled as a source of genetic material. Alright, so they designed a cute little experiment where your virus, it's been labeled so that the DNA is labeled with one radioactive isotope, and the proteins of the virus are labeled with another. And other components of the E. coli bacterium are labeled. Alright. So, at the end of this experiment, they conclude that DNA is injected into your host bacterial cells and becomes a component of the new phages following DNA replication. Serves as a source of genetic information.
In 1953, Alfred Hershey and Martha Chase confirmed DNA's preeminent role in genetics by demonstrating that DNA is the genetic material of a virus called phage T2. The phage, which infects E. coli, consists of a head, sheath, tail, and base plate made of different proteins. DNA is packaged within the head of the virus. When T2 comes in contact with E. coli, the phage attaches to the bacterium by its tail. Next, the phage injects genetic material into the cell. The genetic material directs bacterial enzymes to produce viral offspring. When the life cycle is complete, 100 to 200 progeny phages have been assembled inside each bacterium. The bacterium breaks open, or lyses, and the phages are released. Since T2 consists of only DNA and protein, Hershey and Chase reasoned that the genetic material must be one of the two components, so they designed an experiment to determine which it is. For their experiment, the scientists prepared T2 phages that either had radioactive DNA or radioactive proteins. To make the components radioactive, Hershey and Chase infected E. coli with T2 and grew the bacterial cells in two different culture media, one containing the radioactive isotope phosphorus-32 and the other containing the radioactive isotope sulfur-35. All the phages released from the bacteria grown in the phosphorus-32 medium had radioactively labeled DNA, while all the phages released from the bacteria grown in the sulfur-35 medium had radioactively labeled proteins. In the next phase of their experiment, Hershey and Chase infected E. coli with the two types of labeled T2. They then used a blender and a centrifuge to separate the protein parts of the phage, called the phage ghosts, from the rest of the infected cell. The scientists discovered that the labeled DNA appeared in the host cell, but not the phage ghosts, while the labeled proteins appeared in the phage ghosts, but not the host cell. In addition, the progeny of the T2 with labeled DNA were radioactive, while the progeny of the T2 with labeled proteins were not radioactive. Because genetic material is passed on from parent to offspring, Hershey and Chase concluded that DNA had to be the genetic material of T2. Their results helped convince the scientific community that DNA was the hereditary material.
Alright, so this is a little overview of their experiment. Alright, so they have labeled the protein coat of this virus with a radioactive sulfur compound, isotope. And since proteins have sulfur in them and DNA doesn't, this is a way to just look at proteins. They also labeled radioactively the DNA inside the virus. So they radioactively labeled it with a radioisotope of phosphorus, right? Because you're not going to find a lot of phosphorus in your proteins. All right, so you have your phosphate groups in your DNA. All right, so they went on to allow these phages to infect bacterial cells, E. coli. And then they placed these samples in blenders, and the blenders function to basically detach the phage from the surface of the bacterial cell. Alright, so then they can look at just the phage and look at the bacterial cell. Alright, so here in your protein, your phage proteins are going to be found in the supernatant. Alright, following this centrifugation process. And the proteins are the remnants of the phages, alright, because they remain attached on the outside surface of the bacterial cells, not going to be internalized. And so here, your phage is labeled, but your bacterium isn't. Okay. But in the other experiment, your DNA labeled phages. Alright, the DNA is found inside of the bacterial cell and not in your phage ghost. Alright, so this told them that, okay, the source of genetic information is the DNA and not the proteins. And then when your new progeny developed, alright, the new phages developed and burst out of the bacterial cells, the fact that these phages had DNA in their nucleus that was radioactively labeled, but the phage proteins were not radioactively labeled, also served as substantiatory evidence to support the fact that DNA is a source of genetic material. Alright, so now we know that DNA is a source of genetic information. Alright, we have evidence to support that. But scientists didn't really know what DNA looked like at the molecular level. Alright, they knew that DNA is a polymer, and they knew that this polymer is composed of nucleotides, and then a nucleotide is composed of a nitrogenous base, deoxyribose sugar, and a phosphate group. Alright, so Erwin Chargaff. Alright, through his experiments determined that, okay, DNA composition from one organism to the next varies. And that the diversity of the DNA composition, again, gives more credit to this idea of DNA being a source of genetic information. Now, he also came up with these rules that in a molecule of DNA, the amount of adenine is equal to the amount of thymine. Alright, so again, you should recall complementary base pairing when we talked about the structure of DNA previously. Alright, so it makes sense. The amount of adenine equals the amount of thymine in the molecule DNA. Alright, because for every adenine molecule, we have a complementary thymine nucleotide, or base. And that the amount of guanine in the DNA is equatable to the amount of cytosine, because for every guanine base, you also have a complementary cytosine. Alright, so you should also recall that adenine and guanine are purines, and thymine and cytosine are pyrimidines. Alright, so let's think about this. Using this rule, let's say that Let's say that 80% of molecule DNA is composed of guanine and cytosine. Alright, so that means that you have 40% guanine and that you have 40% cytosine. Alright, so that means you're left with 20%. Alright, so that means that 10% of it is adenine, and the other 10% is thymine. Alright, so looking again at the structure of a nucleotide. Alright, so you should recall that a nucleotide is composed of a nitrogenous base. Alright, in this case we're looking at DNA nucleotides, so deoxyribose sugar and a phosphate. Alright, so without the phosphate, alright, the base and the sugar are referred to as a nucleoside. Alright, so you should also recall your purines and your pyrimidines and which base belongs to each. Alright. Now, again, recall the structure of DNA. Alright, so here we're only looking at one strand of the DNA molecule, one side of it. Now, here, your molecule DNA is composed of your nucleotides, and these nucleotides are added and attached in a specific manner. Alright, there is a directionality to the molecule DNA, alright, to the strands of DNA that make up the molecule. Alright, so one end of the strand is referred to as the 5' end. Well, why is that? Well, it's referred to as the 5' end because you have this free 5' phosphate group that's attached to the 5th carbon of your deoxyribose sugar. Alright, so that's a free 5' phosphate. At the other end of the molecule, the other end of the strand, you have a free 3' hydroxyl. Alright, so the 3' hydroxyl is attached to the 3rd carbon of deoxyribose sugar. You have a free 3' hydroxyl. So one end is 5', the other end is 3'. Now, your DNA nucleotides are always going to be attached in the 5' to 3' direction. Alright, so here you have your little phosphodiester bond linkage. Alright, between your pre-existing nucleotide chain and a new nucleotide. Alright, you have this phosphodiester linkage that holds the nucleotide to the pre-existing chain.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The fourth base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Alright, so here you're being shown complementary base pairing between the 2 strands of DNA polynucleotides that make up a molecule of DNA. Alright, so notice that the 2 strands, alright, they are oriented in opposite directions. Alright, so the term for this is anti-parallel. Alright, so you should recall that. So basically, one strand is going in one direction, the other strand is going in the opposite direction. Alright, so one's oriented 5', 3' in one direction, the other's oriented 5', 3' in the opposite direction. Now, this orientation to the molecule of DNA is important because it kind of gives you a clue to the method by which the molecule DNA replicates. Now, the DNA molecule is actually going to be in this helical structure, so it intertwines on itself. Alright, so a right-hand twist. Alright, so this is showing you the complementary base pairing, the hydrogen bonding between your bases. Alright, so guanine and cytosine having 3 hydrogen bonds, and adenine and thymine having 2. Now, scientists are racing towards discovering the molecular structure of DNA and how the structure ties in with how, in fact, it's inherited. Alright, so here you have Wilkins and Franklin, and they used a technique called X-ray crystallography, alright, to basically look at the molecular structure of DNA. So through this X-ray crystallography, Franklin produced this image of DNA, molecular structure of DNA. Alright, so she discovered that DNA is structured as a helix. Alright, so she basically passed X-rays through the DNA. Alright, basically she crystallized the DNA molecule and passed X-rays through it. And then just like this X-ray is going to be transferred to a plate, to paper, alright, you get an image. And this image showed this little X crisscross structure. And here, this X structure is basically like looking down the molecule of DNA, as if you're standing over it. And she's seeing these strands of DNA are intertwining and passing one another in this X-shaped structure, crisscrosses. Alright, so this led her to come up with this idea that DNA molecule is actually helical in structure. Now, Watson and Crick basically used Franklin's X-ray crystallography image, and they actually obtained this image kind of in probably the most not necessarily ethical manner. Alright, they went to kind of the head of the lab and obtained Rosalind Franklin's X-ray crystallography data and used this in their approach to designing the molecular structure of DNA. And in their paper, in their classical paper on the structure of DNA, Rosalind Franklin basically gets like this end note at the end saying, basically thanks. Alright, so also this is kind of a pioneering time for women in the sciences. Alright. And, at the time, women weren't as accepted in the sciences as men. Alright, so they used this X-ray crystallography data and they applied Chargaff's rules to come up with a molecular structure to figure out how the bases interacted with one another. Alright, and whether or not the phosphates and the deoxyribose sugar and the nitrogenous bases and phosphates, how all these things were interacting, how the structure was arranged. Alright, so they built various models of this helix. And Franklin, through her studies, deciphered that the backbone structure, the sides of the ladder of the molecule DNA, are composed of alternating phosphate sugar residues. And that along the interior, you have your nitrogenous bases. Alright, and then Watson built the model in such a way that the strands were oriented in opposite directions. Alright, it's anti-parallel in nature. Alright, so originally they thought that the bases paired, for example, A with A. Alright, but again, you're trying to put an adenine with another adenine molecule through hydrogen bonding, and two purines together just isn't going to work. Alright, it's too wide. Alright, there's not enough not enough space for these hydrogen bonds to interact and form. Alright, same thing, if you try to place a pyrimidine with a pyrimidine, alright, it's too far apart. Again, no real interaction between hydrogen bonds. A purine and a pyrimidine do work. Alright, so here adenine pairs with thymine, guanine pairs with cytosine. Alright, so again, this molecular structure explains Chargaff's rules. Alright, because Chargaff concluded that the amount of adenine is equatable to the amount of thymine. The amount of cytosine is equatable to the amount of guanine. But he didn't know how that tied in with the molecular structure. Watson and Crick figured out, hey, it has to do with the actual base pairing between these bases. and that you may base pair a purine and a pyrimidine. And this accounts for Chargaff's rules. Alright, they, again, substantiate it. Alright, so this is their molecular structure of DNA. Alright, here's, again, another image showing you the helical nature of DNA and hydrogen bonding.
DNA consists of two complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the two sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. The double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar phosphate backbones.